Okay, so this is my presentation of Chapter 17 on transcription, RNA processing, and translation, which are the processes involved in gene expression using a gene to make a protein or a gene product. The first process is transcription. In terms of regulating gene expression, controlling which genes are used to make proteins when, transcription is the most important process out of the three. So it's actually necessary for having different cell types to be able to regulate gene expression, right? Every cell in your body has the same DNA inside of it, so it has the same genes, but of course the different cells are different from each other. Your skin cell is not the same as a brain cell or a muscle cell or a bone cell. All of them are different. They're able to be different even though they have the same DNA because they do gene expression differently, or they regulate gene expression differently. So each cell type is going to be using genes that it needs for its jobs and not genes that are needed for other cell type jobs. So your skin cells are not going to be using the genes for the brain cells, even though they have them. That is all accomplished using the regulation of gene expression. And transcription is super important for that. So in transcription, on a basic level, you're going to be taking your DNA, separating the two strands of the double helix, so you get this kind of straight separated region, and then you're going to use one of those strands to make a molecule of mRNA. You're only going to use one strand. You do not use both strands or just either one. It's only one every time, the same one every time. So the strand that you actually use to build the RNA is what we call the template strand. You use it as a template to tell you which nucleotide needs to come next in the mRNA that you're building. So here the RNA is being built using the DNA template strand, and here a new nucleotide is coming in. It's an A, an adenosine, so that's going to be able to base pair with this T, and it'll be added to the RNA strand. So it's the sequence of this DNA template strand that is determining the sequence of the mRNA that you build from it. The other strand is called the coding strand, and it actually has the same sequence of bases as the mRNA is going to end up having. That's why it's called the coding strand. It's also sometimes called the complementary strand or the non-template strand. But the coding strand kind of makes sense because its sequence is going to end up being the same as the mRNA. Because, of course, it has to be able to base pair with this DNA template strand, which is the same thing that the mRNA is base pairing with. You always have three phases of transcription: initiation, elongation, and termination. We're going to go through each of those. Transcription is different in bacteria and in eukaryotes, so we're going to talk about it in bacteria first, and then we'll talk about it in eukaryotes. So, first, bacteria. Okay, so in transcription initiation, that's the first phase of transcription. The basic thing that needs to happen there is the polymerase, the RNA polymerase that will build the mRNA, needs to grab the DNA. So RNA polymerase is the enzyme, shown right here, that is going to actually be reading the DNA and using it to build mRNA. And the first thing it needs to do is just grab on. It's actually not very good at grabbing DNA, so it needs some help in the transcription initiation phase. The thing that helps it is the promoter and the sigma protein. So a promoter is a stretch of DNA that is found in front of every gene. So here you have your gene. This is the DNA for the gene. On this diagram of the DNA, this would be your DNA of the gene right here. And then here you have the promoter in front of the gene. You're always going to have the polymerase binding at the promoter first, and then once it grabs the promoter, it will move onto the DNA and start making mRNA. So if it's able to grab the promoter, then you will have mRNA made, and the gene will be expressed. If it cannot grab the promoter, then you will not have mRNA made, and the gene will not be expressed. It will not be used to make protein. Just FYI, we always number the nucleotides in a gene and the promoter using the same scheme. The very first nucleotide in the gene is always the +1 nucleotide, and then the numbers get bigger from there, so +2, +3, +4, so on and so forth. That's all considered downstream from the very beginning, the +1 site. If you go backwards the other way, that's considered moving upstream, and that's where the promoter is. And there the nucleotides would be numbered using negative numbers. So this last nucleotide of the promoter would be -1, then -2, -3, -4, so on and so forth. Within this promoter, you have a couple of regions that are really important for the RNA polymerase binding there. You have the -10 box and the -35 box. Those are each centered on the -10 and -35 nucleotides respectively, and they always have the same sequence. So the -10 box is always TAT-AAT, and the -35 box is always TTG-ACA. Always. Then you're going to have the sigma protein come in and grab the -35 box and the -10 box. It's able to bind those two boxes, and the sigma protein binds the RNA polymerase. So here's kind of a different representation of that, where here you have the RNA polymerase in green. In red, you have the DNA with the two strands shaded differently, and then in blue, you have the sigma protein. So the sigma protein is going to be binding the promoter. The -10 box would be around here, the -35 box is around here, and then the RNA polymerase binds to sigma. Which way the RNA polymerase is pointing whenever it binds to sigma determines which strand is going to be used as the template strand and which strand will be used as the coding strand. For each gene, it's always used the same way. So the template strand is always the same in every single, well, for a given gene, every time that it is transcribed to make mRNA, you always use the same strand as the template strand. You never switch which strand is used as the template. If you did that, you would get a completely different mRNA and a completely different protein out of it. Once the polymerase is grabbed to sigma and sigma has grabbed the promoter, then the polymerase can actually get onto the DNA itself. And then we'll move to the next phase. Sorry, not yet. Okay, so in order for RNA polymerase to actually grab onto this, this stretch of DNA, it's going to need to split open the two strands from the helix. And it will end up feeding them through it. The template strand will be the one down on the bottom, and you're going to have nucleotides feeding into the RNA polymerase. They're going to base pair with the template strand, and the RNA polymerase is going to connect those nucleotides together by catalyzing the formation of phosphodiester bonds between them. And your mRNA will end up feeding out of the RNA polymerase whenever you enter elongation and you actually start building the gene as mRNA. So this little bubble here, where you have the two strands opened, that's called the transcription bubble. The RNA polymerase, as it moves, is going to be opening the two strands in front of it and closing them back up behind it. So it's going to be making this transcription bubble and moving it along with itself as it moves along the DNA. The RNA polymerase has one big limitation, which is that it can only add to the 3' end. So when it has its little bit of mRNA that it's building here, the 5' end is going to be over here on the left, and the 3' end will be over here on the right, pointed downstream towards the rest of the gene that it's going to make. It can only add at the 3' end. So nucleotides have to come in and add here. It cannot add them here. It's impossible. It can only catalyze a phosphodiester bond between the 3' hydroxyl and the 5' phosphate of a new nucleotide. It can't do it the other way around. The energy source for this, of course, you do need energy. You're building a big complicated mRNA molecule, so it's not free, it costs energy. The energy source is not ATP exactly, but it's actually the nucleotides themselves. Every nucleotide has 3 phosphates attached to it. So if you break off the last phosphate, you're going to release energy. Actually, you have to break off 2 phosphates to get the nucleotide to attach to another nucleotide for a phosphodiester bond. So free nucleotides always have 3 phosphate groups on them. Nucleotides that are part of DNA or RNA only have 1 phosphate group. So the RNA polymerase is going to be taking off those last 2 phosphates and getting energy out of it that it uses to actually build the mRNA molecule. So the the RNA polymerase is going to make, just a little bit of its mRNA. It's going to kind of get started, and then it will like to continue moving further down the gene. But the sigma is getting in the way at this point. Sigma is still bound to the promoter and bound to RNA polymerase. So RNA polymerase cannot move along the gene until it lets go of sigma. So at the very end of transcription initiation, the very last thing that happens is that the RNA polymerase releases sigma. So this actually we're going to skip. Yeah, we're just going to skip this. The regulation of transcription initiation is pretty complicated. It's different for bacteria and for eukaryotes. It's very important for gene regulation. It's very important for gene regulation. This is really what determines which genes your cells are using and which ones they are not using. But it's kind of complicated, so we're just gonna skip it this semester. And we're gonna skip this. Okay, transcription elongation. Transcription elongation is the next phase of transcription. It's the phase where you basically build the whole mRNA molecule. It starts when sigma releases the when sigma releases the RNA polymerase. So you have your RNA polymerase. Again, it can only add to the 3' end. It's going to begin moving along the DNA, walking along the DNA. As it goes, the DNA will feed into RNA polymerase. It's going to separate the 2 strands from each other, and then join them back up together when it exits the other side of the RNA polymerase. So this is your transcription bubble that's going to be moving along with the polymerase itself downstream. Nucleotides will come in. They're going to be added to the 3' end of this growing mRNA. They're going to base pair. They're going to base pair with the template strand. Then the polymerase will catalyze phosphodiester bond formation between that new nucleotide and the 3' end of the mRNA strand that it's building. The strand, as it gets longer, is going to feed out the back end of the polymerase. The energy source, again, is the extra phosphates that are attached to each of these nucleotides. So this process is just going to continue like this until you get to the end of the gene. The mRNA will get longer and longer until you get to the end of the gene. Once you get to the end of the gene, then you have the transcription termination phase, where basically you stop transcription. So the way that works in this case is actually you're going to go a little bit past, in bacteria, you're going to go a little bit past the end of the gene. If we say the gene ends maybe right here, you're going to keep going a little ways. But the last part of the gene itself will be the transcription termination signal. When the polymerase transcribes that into the mRNA is going to fold up. So the transcription termination signal has a sequence that's actually complementary to itself, so that in certain places, well, where this neck region is, so that once you have transcribed that into mRNA, that's only one strand, the strand will fold up and base pair with itself and make this little hairpin loop. This hairpin loop gets in the way of the polymerase and actually will push it off of the DNA. And then you'll just have your finished mRNA. Maybe this teensy bit like down here won't be even part of the gene, it will just be extra. And then here's your DNA, and the polymerase will be detached from the DNA. So the hairpin loop here is going to force the polymerase off of the DNA, and then when that happens, the RNA will just kind of float off of the polymerase. So that's one very common way that bacteria stop termination. There's also other mechanisms, other ways of doing it, but we're not going to talk about those. And that is transcription in bacteria. Now we'll talk about transcription in eukaryotes. But again, we're going to skip how transcription initiation is regulated, basically. So it's pretty similar. Well, transcription initiation is pretty similar in eukaryotes as in bacteria, but it's a bit different. So you still have a promoter. The basic thing that needs to happen is that your RNA polymerase needs to grab the promoter so that it can get onto the DNA. But how it grabs is a bit different. In bacteria, you have sigma. You would have a sigma protein that binds the promoter and then binds RNA polymerase. In eukaryotes, it's not like that. You don't have sigma. You have transcription factors instead. Also, you don't have a -10 box or a -35 box in the promoter. Instead, you have the Tata box at -30. You can also have other things in the promoter. Promoters in eukaryotes are extremely diverse. So you're going to have one transcription factor come in and bind the Tata box, and then you're going to have other transcription factors usually also. Different genes will have different numbers of transcription factors that they need to have bind the promoter. It gets actually extremely complicated. So there's any combination of transcription factors that can bind to different parts of the promoter and recruit the polymerase. So once you have all of these transcription factors bound to the promoter, then the RNA polymerase will actually come and grab the transcription factors and be able to get on to the DNA. Your sex hormones, estrogen and testosterone, are actually examples of transcription factors. So estrogen and testosterone are responsible for secondary sexual characteristics. That would be like how men are taller and have bigger muscles, but women have like less body hair. So every cell in your body pretty much is affected by those sex hormones, and those sex hormones are transcription factors. So they go into the DNA in the nucleus and they bind promoters and determine where RNA polymerase is going to be able to grab onto the DNA and which genes it's going to be able to transcribe. So this system of having a bunch of different transcription factors is very complicated, but it's good because it allows very sophisticated gene regulation and a lot of diversity in the cell types. Right, so we're going to skip this, and we're going to continue with this. So, as I didn't mention, but again, transcription initiation ends when the RNA polymerase lets go of the transcription factors, and at that point, transcription elongation begins. Transcription elongation is basically the same in bacteria and in eukaryotes. The only difference is that in eukaryotes, the DNA is actually wrapped around histone proteins, and you have to move the histones out of the way. Other than that, it's just the same. Then you have transcription termination. The way that transcription stops in eukaryotes is pretty different from how it stops in bacteria. So in bacteria, you will commonly have the transcription termination signal transcribed into the mRNA, and then that forms a hairpin loop in the mRNA, which forces the polymerase off of the DNA. In eukaryotes, it is different. In eukaryotes, here you have your RNA polymerase chugging along, transcribing the DNA. Here you have the mRNA strand that it is building, and then here is the tail of the polymerase. So every RNA polymerase in eukaryotes that does transcription is going to have this little tail coming off the back of it, and it has enzymes on that tail. So you're going to at some point transcribe a poly A signal in the DNA. So as this polymerase is moving down, it moves past the poly A signal, ends up transcribing that, and that comes out in the mRNA here. And then the enzymes on this tail of the polymerase are going to recognize that little stretch of A's and cut the mRNA right there. So that now here you have your mRNA. This is actually shown as being processed already, but here would be your poly A signal that has stopped the transcription, and then the polymerase itself is going to end up just letting go. So the polymerase will come off of the DNA, and yeah, then it's over. I'm not sure why my picture looks like that. Oh well. Okay, after transcription, you have RNA processing in eukaryotes. In bacteria, you do not have RNA processing after transcription, you go straight to translation. But in eukaryotes, you do RNA processing first. There's different ways that RNA can be processed. The main things are adding the cap and the tail and doing the RNA splicing. Oh, where's my laser pointer? Okay, so once you first finish the mRNA, once you finish transcribing that from the DNA, what you actually have is technically called a primary transcript or a pre-mRNA that reflects the fact that it has not been processed yet. When you're processing it, you're going to add a 5' cap and a poly A tail to the other end, to the 3' end. That's the cap and the tail. Every mRNA in eukaryotes has a cap and a tail added to it. And then you're going to cut out little stretches from the middle of the mRNA until you get the mature actual mRNA. That's called Rna splicing. So all of these kind of greenish chunks in the mRNA ultimately are going to get cut out, and you're left behind with only the orange chunks. So those green chunks are called the introns, and the orange chunks are the exons. In RNA splicing, you have enzymes that take the pre-mRNA transcript and cut out all of those introns and join the exons together. So all these little orange chunks are going to end up attached to each other with no green in between, and that's your mature mRNA. There's actually different ways that can be done. Right, so sometimes you might keep one of these introns, sometimes you might get rid of one of these exons. So for any mRNA, there's generally multiple ways that you can splice it to get different mature mRNAs and therefore different proteins at the end. Actually, 95% of human genes undergo alternative splicing so that their pre-mRNAs are spliced differently to make different mature mRNAs and different protein products. That's actually really important for a lot of different genes. And we're actually going to just skip this FOSS example. We're not going to go into the splicing very much. We're going to skip this. It's kind of about the splicing, but we're going to skip it. Okay, here's more about the cap and the tail. So the 5' cap is a methylated guanine that is added to the end, to the 5' end of the mRNA. So that's an unusual nucleotide. It's like a modified nucleotide, and it's added in a weird way. So between this guanine and the rest of the mRNA, you actually do not have a phosphodiester bond. It's a different type of bond. It's a special way of attaching nucleotides to each other. Then at the other end of the mRNA, you're going to have an enzyme come and just add a bunch of A's to the 3' end until you get this poly A tail. The purpose of the 5' cap and the poly A tail are to protect the mRNA from degradation and to help it bind to the ribosome. So here you have in kind of tan here you have a ribosome. As you see, it's very complicated. It has a lot of different proteins that are all part of it. And here's in blue the mRNA that the ribosome is going to grab. So it actually grabs the cap, the 5' cap of the mRNA, and then the whole thing will loop around so that the ribosome can grab the poly A tail again. It needs to grab those things to be able to get onto the mRNA and starts transcribing it into a protein. Translating it into a protein. So that's one purpose of the cap and the tail. Then the other is to protect the mRNA from degradation. So actually, all eukaryotic cells have enzymes called Rnases that continually find RNA and destroy it. And they do that because viruses, if your cells become infected with a virus, viruses will kind of use their own RNA to force your host cell to make proteins for the virus and to make new viral particles. So when a virus gets into the cell, it's going to inject its genetic material into that host cell. The virus has no way to replicate by itself. It has to get what it needs from the host cell, and the virus actually doesn't have the ability to make protein or to make new viral particles. So the host cell has to do that. The virus has to force the host cell to make proteins and new viral particles for the virus. It's going to do that by having its own RNA and having the host cell's ribosomes take that RNA and turn it into proteins. Then the proteins will assemble the virus. So your eukaryotic cells have evolved to consistently destroy RNA when they find it. So when they find some RNA, they just figure it's viral RNA, it's dangerous, and they break it down. In order to be protected from that, RNA has to have the 5' cap and the poly A tail. So all of your own mRNAs will have that cap and tail added to them. That way, when you send them into the cytoplasm with the RNase enzymes that degrade RNA, they're going to be protected and they won't be degraded. Okay, so finally you have translation. In translation, you take your mRNA and convert it into protein. So that's the final stage of gene expression. Translation is different in eukaryotes and in bacteria, but we're going to kind of talk about both of them just at the same time because it's actually very similar between the two with just some differences. So you always have 3 phases of translation. They are initiation, elongation, and termination. So that's basically the same 3 phases that you have with transcription. And you also have 5 components. So you have your mRNA, of course. You have your ribosome that actually builds the protein from the mRNA. You have amino acids that are going to be put together into that new protein. You have tRNAs that are going to bring in those amino acids to the ribosome. And finally, you have rRNA. That's actually part of the ribosome, but it's so important that we're talking about it separate because that's the part of the ribosome that actually does the catalysis to add 2 amino acids together and form a peptide bond between them. So first we'll go over some of those components, the tRNA and then the ribosome. T in tRNA stands for transfer, so that's transfer RNA. Transfer RNA or tRNA always has this shape. The actual 3D shape of it looks kind of like this, but a lot of the time we represent it flattened out like this, so it looks like kind of a cross. On one end you have 3 nucleotides that form the anticodon. That's going to pair up, base pair, with the codon on the mRNA. On the opposite end, you have an amino acid attached. Yeah, so those are your 2 basic things that you have to have on your tRNA, an anticodon on one end and an amino acid on the other end. So your mRNA is going to be divided into these chunks called the codons, which are each 3 nucleotides long. Each of those specifies one particular amino acid, and it's actually the tRNA that kind of determines which amino acid it is. So for every tRNA that has a particular anticodon that would be able to bind with a particular codon in the mRNA by base pairing, on the other end you're going to have one specific amino acid. So for this UGA anticodon on your tRNA, you're always going to have serine attached to that tRNA. And that's what makes the genetic code unambiguous. Once you have the amino acid actually attached to the tRNA, then we technically call it aminoacyl tRNA. So if we want to get technical, just tRNA doesn't have an amino acid attached to it yet. Once the amino acid is added, then it's an aminoacyl tRNA. The enzyme that does that is the aminoacyl tRNA synthetase. You have a number of different ones actually. Every tRNA or every amino acid actually has its own aminoacyl tRNA synthetase. The process of taking an amino acid and attaching it to the tRNA is called charging the tRNA, and it does require ATP. Code redundancy is also determined by the tRNAs. So when you have the anticodon base pair with the codon, it actually doesn't line up quite right. The first two nucleotides line up like perfectly, but because the anticodon is curved, by the time you get to the third nucleotide, it's actually at an angle like this here. So it's not going to be able to base pair perfectly with the nucleotide that it's supposed to pair with. That's called the wobble position because it's kind of wobbly, it's not straight, it's at an angle. Because the base pairing is going to be wonky anyways, sometimes it's possible for there to be a base pair made with your last nucleotide of the anticodon and different nucleotides in the codon. So this G here should be pairing with a C, but because it's at an angle, sometimes it can also pair with a U. And that is why the codon UCC would mean whatever amino acid this tRNA is attached to, and the codon UCU would also mean the same amino acid. So this is what gives rise to the code redundancy and the conservative properties of the genetic code, where you can have different codons all coding for the same amino acid, and the difference between them would always be the last position, the last nucleotide in the codon. Then your ribosome is a really huge protein complex. It contains proteins, many different proteins, and rRNA. Here you have kind of a realistic-esque diagram of the ribosome with all the little bubble bits. Those are amino acids of the proteins in there. And then in yellow, you can see the RNA running through it. That's the rRNA, the ribosomal RNA. That's actually responsible for catalyzing peptide bond formation between two amino acids. Here you have a tRNA coming in there. This would actually be another tRNA poking out here and a third tRNA over here. You have two different, well, there's a lot of components of the ribosome, but they're kind of divided into two big parts, the small subunit and the large subunit. The mRNA is going to end up fitting in between those two subunits. So in our realistic diagram, the small subunit is kind of in tan or beige here, and the large subunit is in gray. Within each of those subunits, you have multiple different little proteins in there, which we're not going to worry about. Then finally, you have three places inside of the ribosome where tRNAs can come in. They're called the A site, the P site, and the E site, or from left to right, that would be EPA. tRNAs are actually going to come into the A site, that's the acceptor site. Then the ribosome is going to be moving down along the mRNA. You're going to have the A site tRNA move into the P site, and at that point, that's called the peptidyl site. That's where peptide bonds would actually be catalyzed. Then as the mRNA moves more, that tRNA will end up in the E site, which is the exit site where it leaves. So A is for tRNAs coming in, P is for peptide bond formation, and E is for amino acids leaving. Sorry, tRNAs with their tRNAs leaving. So here's translation initiation. The first thing that happens is that your small subunit is going to bind ultimately to the mRNA. So here's your small subunit and the mRNA. You'll also have some kind of other proteins come in, the initiation factors, to help the mRNA bind to the small subunit. If we're talking about a prokaryote like a bacteria, then you have a special region on the mRNA called the Shine-Dalgarno sequence, where the ribosome will actually be binding. If we're talking about a eukaryote, the ribosome is going to grab the 5' cap and the poly A tail. Once your mRNA is attached to the small subunit, then you're going to have a tRNA come in and base pair with the start codon. The start codon should be positioned basically in the middle of the ribosome in what's going to end up being the P site. and this tRNA will come in. AUG is the start codon. The tRNA that will base pair with that always has a methionine attached to it. So you always have methionine as the first amino acid of your protein. After that tRNA is there, the large subunit of the ribosome is going to come down on top of everything. So here you have the mRNA finally sandwiched between the two subunits of the ribosome, and you have a tRNA positioned in the P site and base pairing with the start codon on the mRNA. Once the ribosome is assembled onto the mRNA like that, then you switch from translation initiation to translation elongation, which is basically the same in prokaryotes and in bacteria, in bacteria and in eukaryotes, or prokaryotes and eukaryotes. So you kind of start with this tRNA in your P site, then you're going to have another tRNA come into the A site, the acceptor site. It's going to have its anticodon base pair with the codon at that site, and it brings whatever amino acid is attached to it. Then you're going to have the ribosome spend ATP in order to form a peptide bond between the amino acids on the tRNAs and the P and A sites. It would be the rRNA that actually does that again. Once that happens, you're going to have the tRNA in the P site actually detached from its amino acid, so this whole little amino acid chain is going to be transferred to the A site tRNA. Then the ribosome is going to move along the mRNA. It's going to walk along the mRNA. When that happens, whatever was in the P site ends up in the E site. Whatever was in the A site ends up in the P site. The tRNA in the E site just leaves because that's the exit site. Then you have this tRNA left over in the P site, and the process is going to repeat where you have a new tRNA come into the P site, bring in its amino acid. That's going to have a peptide bond formed with the amino acid chain. The chain will be transferred to the A tRNA, the A site tRNA. Then the ribosome is going to walk, using ATP to walk along the mRNA. Your tRNAs will shift down one site, and whoever ends up in the E site is going to leave. That process is going to repeat until you've walked along the entire mRNA. Even though this happens very similar in prokaryotes and eukaryotes, in eukaryotes you're also going to have a number of elongation factors, which are extra proteins that need to come in and attach to the ribosome and the mRNA so that the ribosome can stay on it without falling off. Then, once you get to a stop codon towards the end of your mRNA, you'll have translation termination. So your three stop codons are UAA, UAG, and UGA. If you find any of those sequences as codons anywhere in the mRNA, the ribosome is going to stop. Once your stop codon is positioned under the A site, you're going to have, instead of a tRNA coming in, you're going to have a release factor protein come in. It's shaped very similar to a tRNA, and it can actually attach to the stop codon, to any of those three stop codons. But since it's not a tRNA, it doesn't have an amino acid attached to it. So then, when your ribosome goes to try to attach your amino acid to the new amino acid, there is no new amino acid, so it doesn't work. But you do still break the bond between this P site tRNA and the amino acid it was attached to. So that means that your chain of amino acids is now detached entirely from the tRNA and from the ribosome. It's just going to float out. And at that point, everything will basically disassemble. Your tRNAs that are still in there are going to leave, and the large subunit will come up off of the small subunit so that the mRNA floats free as well. At that point, either you can degrade the mRNA or you can reuse it to make a new protein from it. Usually an mRNA is going to be reused multiple times to make multiple copies of the protein. One really important difference between transcription and translation in bacteria and in eukaryotes that we didn't talk about is the location in the cell where these things are happening. because eukaryotes have their DNA in a nucleus, and prokaryotes just have it in the nucleoid, part of the cytoplasm. So in your eukaryotes, you have transcription in the nucleus that makes mRNA. The mRNA has to be processed, then it has to be exported out of the nucleus to the cytoplasm, and there it'll find a ribosome and you'll translate it into a protein. In prokaryotes, since there is no nucleus, you can have transcription and translation happening in the same place in the cell, and that means that it can actually happen at the same time. So here, you can see, like, well, it's kind of faint, but there's a little line right here. That is actually a line of DNA in the nucleoid of a bacterial cell. All of these little dots coming off of it are ribosomes. And then wherever that little line of ribosome dots connects to the DNA strand, there's a smaller little dot. Right there, right there, and right there, and right there. That small little dot is the RNA polymerase. So you're going to have RNA polymerase moving along this DNA, transcribing it to mRNA, and as it goes, it has a chain, like a trail of mRNA, a tail of mRNA coming out of it. As it moves, that tail is going to get longer and longer, and ribosomes are going to come and attach to it and start making amino acid chains or proteins based on it. So each of these little lines or little trails of dots is an mRNA that is being translated into protein by multiple ribosomes simultaneously. If you make a diagram of that, it looks like this. Here is your DNA that's being transcribed by each of these RNA polymerases. So you're in the process of making 4 mRNA transcripts right here. Here's in red the mRNA that's coming off of each RNA polymerase as it's being made. Before that mRNA is even done, you can have ribosomes attaching to it because the ribosomes are in the same place as the DNA, and those ribosomes will start making amino acid chains based on it. So by the time this RNA polymerase actually gets to the end of the gene, you're going to have ribosomes that are just about done making it, translating it into protein. This allows prokaryotes to make a lot of protein very quickly from their DNA. In eukaryotes, it takes longer. It's a slower process where you have to process the mRNA and export it out of the nucleus, so you have to finish transcription before you start translation, and in prokaryotes, everything happens at the same time. So after translation, we're pretty much done, but we do have a few more things that can happen after translation to those proteins that we just made. Of course, they need to fold up. Is one of the first things that happens after translation. The protein won't actually be functional until it has finished folding, usually in the rough endoplasmic reticulum. After the protein is folded, you can also add stuff to it. You can add different chemicals to it. There's a bunch of different options for what you can add to proteins in the rough endoplasmic reticulum and in the Golgi apparatus mostly. Let's see, here you're adding a hydroxyl group to this protein, that's hydroxylation. In lipidation, you're adding a lipid to it. In phosphorylation, that one's real common, you're adding a phosphate group to it. In methylation, you're adding a methyl group to it. So all of these are different modifications you can make to the protein. Those modifications will change. Well, here in glycoscalation, actually you're adding a sugar to it, an oligosaccharide to it. All of those modifications are going to change something about the protein. Maybe where it goes to in the cell, maybe what its function is when it gets to its destination. Maybe how long it lasts. This one right here, ubiquitination, adding ubiquitin. It's a tiny little protein that marks, well, it's a tiny peptide actually. It marks the protein for degradation actually. So when a protein is old and it needs to be broken down, you attach a ubiquitin to it and then it gets broken down. So all of those modifications mean something for the protein in terms of how long it will last, where it goes, or what its function is going to be. So the proteome is all of the proteins that are in a cell. And it's very diverse. Cells can make a lot of different proteins. You start, of course, with your genome. The genome is all of the DNA inside of the cell, or all of the genetic information inside of the cell. In a human cell, we'll have about 20 to 25,000 genes. Well, I guess I should say in a eukaryotic cell. Well, in an animal cell. In an animal cell, we'll have about 20 to 25,000 genes. But the final amount of proteins in the proteome is over 1 million. So you're getting a lot done with only a few genes comparatively. It's a massive difference. Let's see. That's 2 orders of magnitude different between how many genes you have and how many proteins you can make. So first you have alternative RNA splicing, so that for each of these genes in your genome, you're going to get a pre-mRNA off of it that can be spliced in different ways to make several different versions of that mRNA that will each turn into a different protein. Then once you've done the translation and you're going to modify those proteins, you can modify the same protein in different ways, and that will change things about the protein, like where it goes to and what it does. So that adds a lot to the proteome as well. So you go from about 25,000 genes, then after alternative splicing, you end up with about 100,000 different mRNA transcripts, and then after the post-translational modifications, you'll end up with over 1 million different proteins in your cells. And that's that. Oh no, summary, haha. Okay, so the whole summary for gene expression in a eukaryotic cell here, because we have a nucleus. So we start off in the nucleus, we do transcription, you have RNA polymerase, it initiates by attaching to the promoter, in eukaryotes using transcription factors, in prokaryotes using sigma. Once it lets go of the transcription factors, or if sigma is going to enter the elongation phase, move along the DNA and make the mRNA as it goes, technically pre-mRNA, once it gets to the end of the gene, it'll stop. In prokaryotes, you'll have the transcription termination signal making a hairpin loop in the mRNA that pushes the RNA polymerase off of the DNA. In eukaryotes, you'll have a poly-A signal, and then the little tail on the RNA polymerase will cut the mRNA transcript at that signal. But however it happens, you terminate transcription and you're left with your pre-mRNA transcript, or your primary transcript. It is not processed yet. In eukaryotes, well, this would only be in eukaryotes processing. In eukaryotes, you're going to add the 5' cap to it and the poly-A tail to it. Those are going to help it attach to the ribosome later and also protect it from degradation. And you're finally going to cut out the introns and join together the exons. That's RNA splicing, and there's different ways that you can do that to make different mRNA transcripts. Then you're going to export that mRNA outside of the nucleus into the cytoplasm, where it's going to find a ribosome. The ribosome is going to attach to the mRNA. How it grabs it is a little different between prokaryotes and eukaryotes. In prokaryotes, it's going to grab the Shine-Delgarno sequence on the mRNA itself. In eukaryotes, it's going to grab the 5' cap and poly-A tail. Once the ribosome assembles, you're going to have your tRNAs coming in, bringing in amino acids, moving from the A site to the P site to the E site, and then leaving. Each time a new tRNA comes in, it will base pair with the codon, and its amino acid will be added to the chain by rRNA in the ribosome. Once, I guess that would be your translation elongation phase. Once you get to the end of the mRNA, you'll have the translation termination, where you reach a stop codon and the release factor protein comes in and everything lets go of everything else. Then finally, your protein, your amino acid chain is going to fold up to make the right shape, and it'll be modified in different ways to affect its location, destination, location and function.